Today's topic for discussion is life cycle of bacteriophages. The coliphage particle is carried close to the coli cells. That means the viruses which infect E. coli are described as coliphages, and the tail plate and the caudal fibers anchor the phage viron to the host cell wall at a specific site which the tip of the phage tail happens to touch. With the attachment of the phage to the specific receptor site on the host cell, start the life cycle of the phage. The next step is the injection of the phage DNA, and within a few seconds, a opening is formed in the host cell wall at the point of attachment by the enzyme in the tip of the tail. The elastic protein sheath of the tail now contracts, driving the hollow core into the host cell like hypodermic syringe through the opening. The phage DNA is then forced down to hollow core and is injected into the host protoplasm. The protein coat of the phage remains sticking as an empty shell on the outside. The steps opened the above continues to absorption and penetration phase and infection phase in the life cycle of bacteriophages. Next follows the latent period phase, or it is also known as period of eclipse. What happens during this period? During the eclipse period, which is usually about 12 to 22 minutes, no virus DNA as such can be seen in the host cell. It disappears as an independent infective agent. It seems that the phage DNA within the host cell enters the nuclear or chromatin body of the host cell and takes control of the cell's genetic machinery in a manner unknown at present. The replication of bacterial DNA and synthesis of bacterial enzymes occurs immediately under influence of phage DNA. The chromatin body of the host, which also contains DNA, is broken down and its content is dispersed within the first few minutes after infection. The bacterial enzyme system and either commanded or converted into systems capable of elaborating phage components from the cell content and integrants absorbed from the medium. And the phage DNA is replicated in the region of the nuclear or chromatin body of the host cell. The host cell's ribosomal factory in cytoplasm is caused to elaborate new phage coat proteins. It produces new proteins. The next step in the phage life cycle is the assembly of the phage component into a new phage particles. As the elaborated particles of the phage DNA leave the chromatin region of the host cell, the new phage protein quickly aggregate round them to constitute new stable virons of T1 phages. Assembly of phage units starts about 12 to 22 minutes after infection. The tail is finally added to the phage head. Within about half an hour after infection, the host cell contains 2 to 300 or even more particles of T1 phages or bacteriophages. The period of production of new phage particles is called vegetative phage. It comprises a replication of DNA, synthesis phage protein, and assembly of phage components into new phage particles. The final steps in the phage life cycle is the liberation phage. The host cell or whatever is left of it suddenly bursts to release the phage progeny or the bacteriophages. The process of disintegration or destruction of coliphage cell is called lysis. The phage which brings about lysis or destruction of the host bacterium is called a virulent phage. This is all about life cycle of bacteriophage or multiplication of bacteriophage. Thank you very much. Be clean, be safe.